See. What is the relationship between psilocybin, neuroplasticity, and social belonging? In recent years, psilocybin has become one of the most important compounds in neuroscience research because it appears capable of temporarily reorganizing large-scale brain networks while also producing long-term psychological effects. What makes this especially interesting is that many people who undergo psilocybin experiences report increased feeling of emotional openness, empathy, and social connectedness. I feel attracted to this topic because in some countries of Latin America, we consume eventually as a ritual in ceremonies with guidance to heal the mind, and it is far from being addictive. The central question is, how can a pharmacological compound alter brain architecture in a way that changes not only cognition but also our perception of belonging and interpersonal connection? Today, I will explain this through three main ideas. The first one, as you can read, how psilocybin changes large-scale brain networks. Second, how it induces neuroplasticity. And third, how those neural changes may increase social connectedness. Psilocybin is a natural psychedelic compound found in certain mushrooms species. Once ingested, it is rapidly converted in psilocin, which primarily acts on serotonin 5-HT2A receptors. These receptors are concentrated in cortical association regions involved in self-awareness, emotion, and high-level cognition. Psilocybin appears to affect the overall organization of brain activity. This is why researchers increasingly study psilocybin not simply as a chemical that changes mood, but as a compound capable of altering network-level brain dynamics. The region most affected include the prefrontal cortex, which regulates executive control, you know that, the posterior cingulate cortex, which is heavily involved in self-referential thinking, the insula, which processes interoception and emotion, and finally, the limbic structures, as you know, are involved in affective processing. To understand psilocybin, we first need to understand how the brain is organized. I know you know that, but I like to remember that. The brain is not a collection of isolated regions. Instead, it operates through large-scale functional networks. One of the most important is the default mode network, or DMN. As you mentioned in class, the DMN is heavily involved in self-referential processing that helps maintain our autobiographical narrative and our sense of identity. This is not bad, this is not good. Another important system is the salience network, which determines what stimuli are emotionally or behaviorally relevant. Finally, the central executive network supports cognitive control, decision-making, and working memory. Research from Nature Communications and work by Robin Carhart-Harris, who is a scientist expert in this compound, he shows that psilocybin significantly disrupts the DMN, the default mode network. Under normal conditions, this network acts like a stabilizing system for the self. It maintains our internal narrative and organizes cognition around a coherent sense of me. Under psilocybin, coherence within the DMN decreases. This means that the network becomes less synchronized and less dominant over overall brain organization. At the same time, communication between normally separated networks increases. This disruption is associated with what many participants describe as ego dissolution, meaning a temporary weakening of rigid self-boundaries. Interesting, no? Importantly, this does not mean people lose consciousness. Rather, the structure organizing self-processing becomes more flexible. This flexibility appears central to therapeutic effects and changes in social perception. So here we can see in panel A, this shows the brain network analyzed. In orange is the DMN. This one is associated with internal thought and the sense of self, as I mentioned before. In blue is the salience network, which is involved in detecting and prioritizing important stimuli. Here in panel B, under placebo, the red and blue signals move in opposite directions. This indicates that the networks are anti-correlated, which is the brain typical baseline pattern that happens normally. So here, which is very interesting, is in panel C after psilocybin, both signals begin to move together. This reflects increased functional connectivity between networks that are normally more segregated. Here in panel D shows positive orange and negative blue connections from another key DMN region, which is the ventromedial prefrontal cortex. Finally, in panels E and F show the full brain scan under psilocybin. At the beginning, connectivity is low, but as the effect intensifies, it increases substantially, indicating a stronger synchronization across multiple brain networks. Overall conclusion, psilocybin appears to shift brain dynamics toward a less rigid and more integrated state, increasing communication between networks that are usually functionally separated. One of the most influential theories in psychedelic neuroscience is the entropic brain theory. According to this model, psilocybin increases neural entropy. Entropy here refers to variability and complexity in brain activity. Normally, the brain operates in relatively stable and predictable patterns. Under psilocybin, those patterns become more flexible and less constrained. EEG studies show increased signal complexity and increased LEM-PEL-CIP complexity, which is a measure of how unpredictable neural activity becomes. This does not mean the brain becomes chaotic. Instead, the brain enters a high flexibility state where it can explore a broader range of neural configurations. This increased flexibility may allow the brain to escape rigid cognitive and emotional patterns. Here we can see the modern neuroscience increasingly describes the brain as a predictive system. The brain constantly generates models about reality and about the self. This model help us efficiently interpret sensory information. So the interesting thing is psilocybin appears to reduce the precision of these high-level predictions. This weakens top-down control from higher cortical systems. As a result, sensory and emotional information gains greater influence. This process helps explain why people under psilocybin often experience increased emotional sensitivity, altered perception, and reduced rigidity of identity. Importantly, when the self-model becomes more flexible, interpersonal processing may also become more flexible. The boundary between self and other becomes less rigid, increasing emotional resonance with others. One of the most important findings in recent psychedelic research is that psilocybin may induce neuroplasticity. Here, neuroplasticity refers to the brain's ability to reorganize itself structurally and functionally. Studies discussed in behavioral neuroscience show increased synaptogenesis, the biological process by which neurons form synapses, and dendritic spine growth following psilocybin exposure. Researchers also observed activation of molecular pathways related to brain-derived neurotrophic factor or BDNF, which is strongly associated with learning and neural adaptation. Meanwhile, diffusion imaging studies found measurable changes in white matter microstructure after psilocybin experiences. These findings suggest that psilocybin temporarily opens a plasticity window during which the brain becomes more adaptable to experience. This is extremely important because it means, obviously, the context matters. Social experiences occurring during this plastic state may become deeply encoded. One of the most consistent findings in psychedelic research is increased feeling of social connectedness. Participants frequently report feeling emotionally closer to others, more empathetic, and less isolated. From a neuroscience perspective, these effects may emerge from reduced rigidity in self-processing systems. As the DMN becomes less dominant, the distinction between self and other becomes more permeable. At the same time, emotional and interoceptive systems becomes more integrated. This increases sensitivity to emotional cues and interpersonal information. Research Research from Yale also suggests that psilocybin-assisted therapy may reduce chronic social isolation, commonly associated with depression and trauma. Oof, this sounds beautiful, no? This suggests that social belonging may not simply be psychological, but deeply rooted in the organization of large-scale neurosystems. So in this quasi-final slide, I want to tell you about how integrated systems model works. So first, psilocybin activates the 5-HT2A receptors that are part of the serotonin system. This increases cortical excitability and destabilizes normal network organization. After that, the brain enters a high entropy state characterized by increased connectivity and reduced modular segregation. Uh, this state relaxes rigid predictivity processing and destabilizes self-referential processing. At the same time, neuroplasticity mechanism increase the brain capacity of reorganize. Finally, these changes may produce increased emotional openness, empathy, and feelings of social belonging. In this framework, social connection is not simply a psychological side effect, it emerges from large-scale changes in neural architecture. Overall, psilocybin research provides a unique window into how brain architecture shapes not only perception and cognition, but also the human experience of connection and belonging. So this is my last page about references. You can check it if you wanted to. And I am going to stop